So, in keeping with the great talks that we've had this morning, that we're going to have two wonderful speakers as well this afternoon. And the first one is Dr. David Huntsman, who's a professor in the departments of pathology and laboratory medicine, a department that's close to my heart, of course, and obstetrics and gynecology at the University of British Columbia, and is the Dr. Chu Wei Memorial Professor of Gynecological Oncology. He also works as a staff pathologist at the Cancer Agency and at Vancouver General Hospital, so he's busy. Currently, he's the director of the BC Multidisciplinary Ovarian Cancer Research Team and director at the Centre for Translational and Applied Genomics. And he's got a whole bunch of other things along with that. David's published over 100 papers, and his research has led to the development of predictive and prognostic tissue-based cancer biomarkers for ovarian cancer and a wide variety of other tumors, including sarcomas. He and his team have created a blueprint for subtype-specific ovarian cancer control and have been leaders in the application of novel genomic technologies as applied to ovarian cancer. His talk today, which is paraphrased from William Osler, as goes your genomic medicine, so goes your personalized medicine. Welcome.
Thank you for including me in this, what has been a really enjoyable meeting. A few weeks ago, I received an unsolicited email, which was an advertisement for a book by a physicist, and the review of the book said, the author is only occasionally inappropriately provocative, and that is my goal for this talk. So Sir William Osler was a great Canadian. He stressed lifelong learning and listening to patients. He invented the residency. He wrote the Principle and Practice of Medicine. He was also a quotable humorist, and a lot of the things he said were quite applicable to today's talk. He understood genetics, for instance, and said the best way to prevent varicose veins is to change your grandparents, which was quite clever. And he also said, as goes your pathology, so goes your clinical medicine. So I made this, and I regretted it afterwards, this foolish paraphrase, because then I had to build a talk around it. But I'm not sure about the accuracy of this, and I don't know what the way forward is going to be, but I'm going to present some ideas which may be inappropriately provocative, but I'm hoping at least they may lead to some discussions. So this is a sort of the plot of the story. How we're using cancer pathology today, genomics, as a discovery tool, the concepts of inter- and intratumoral heterogeneity. What are we going to do with a new taxonomy? How can we use it? And then it may seem a little self-absorbed since I'm a pathologist, but I think the who, the where, and the how of personalized medicine testing is important to sort out if we're going to move forward. So cancer is a great disease to study for personalized medicine because it's a disease of mutations which are acquired over time. And when you're studying a cancer, you can compare it to normal. And so it's a little bit easier than a lot of the family studies which have been alluded to. When you look at cancers which we do well with and those which we're really struggling with, they can usually be tracked onto some sort of map which goes from very generic approaches to more stratified approaches to care. At some point, we may offer individualized care, but it may not be necessary for all instances. So this is cancer pathology. In the 1850s, this brilliant idea to use microscopes to look at tumor samples kind of emerged, and the Germans basically classified the world of tumors, and it provided a lot of information. It could tell you roughly what kind of cancer you're dealing with. But where we struggle is to look at a tumor and say, well, how's this going to behave? It's a breast carcinoma, but what's going to happen next? And now, in the past, when there were no treatments available, that really didn't matter, but now there are different options. We can draw some help from things like immunohistochemistry for ER or HER2, but really, it's a bit of a mug's game in the end. It doesn't provide the type of information we really need to move forward with. And I think it's a little bit like looking at something else which grows very rapidly inside of people and then trying to figure out what's going to happen with it, as in babies. And so you look at these baby pictures, and what is their fate going to be? Who's going to be terrible? Who's going to do well? Who no one's ever going to hear of again? And it turns out that this is Ernest Hemingway, a great writer. This is an absolutely evil man, Adolf Hitler, and this is Harry Truman. And I mean, who would you know how would you know that from looking at those pictures? And so we need biomarkers. And in the past, we went one gene at a time. We had HER2, ER, then the BRCA1 and 2 genes, which Stephen in the audience played a major role in identifying. Or you can look at everything once, the genomic approach, which of course started with cytogenetics, very low resolution genomic views of tumors, showing that these are normal cells, these ovarian carcinomas are really messed up. Then expression profiling in the 1990s, which could show you patterns in groups of tumors, but it didn't really provide the resolution to tell you exactly what to do with an individual patient or why that tumor looked that way. And I think expression profiling is a bit like this painting by Turner, where you can see there's something awful going on, but you can't really tell what on earth this thing is. And so these genomic assays which came out, and a lot of them, and I think several speakers have alluded to the fact there's a lot of stuff out there which is kind of pretty shocking, and this is my favorite. It's a microRNA test to tell you whether it's a squamous or an adenocarcinoma, and I believe you can actually teach a chimpanzee how to make that diagnosis, so I don't know why you need to spend thousands of dollars on a special test. And because of this and issues such as the Duke problems with the analysis, the Institute of Medicine came up with a report. And I actually like this report on how genomic assays have to move from a discovery to primary validation, and then only when you have everything locked down do you start actually testing to see, could this work clinically? And it's free online, and it's anyone who's interested in biomarker research. It really kind of lays out a framework for how you could move forward. And it's both terrifying, but it also lays out actionable steps you can actually adopt. But expression profiling really could only take us that far, as I alluded to. We needed more information from that. And this is why I think as a pathologist and a part of the pathology community, everyone was so excited about biology's Man on the Moon feat, which was published just over 10 years ago. And the hope was that we'd suddenly have these tools to better prevent, diagnose, and treat disease, and we'd be able to use different features of disease to monitor therapy. But it was a bit of a challenge because if this was the kind of the Man on the Moon experience, we didn't really move forward very fast for the first few years. There were iterative improvements. And the problem was technical. We could do high-resolution analysis on small fragments of the genome, or we could do a very superficial analysis of everything, but you couldn't do the high-resolution analysis on everything. So if this was the Man on the Moon experience, only very few people could go to the moon. And of course, that's changed. And this is lots of sort of versions of this slide will probably be shown today. And what I like is the sort of fanciful concept, but if you wait long enough, people will pay you vast amounts of money to get your sequence done, but it may not work like that. But basically, we can do research now, which would have been unimaginably kind of foolish to even talk about a few years ago. For instance, we can decode cancers and host genomes and look at the what and why of cancer. We can study intratumoral heterogeneity and start to study in the context of clinical trials and in real time with patients the emergence of drug resistance so we can better handle that problem. We're lucky in Vancouver in that we have an excellent Genome Sciences Center run by Mark O'Mara and Steve Jones, and they opened the door of their Genome Sciences Center to multidisciplinary research teams like the one which I operate so we could get our tumors sequenced. I'm also very fortunate to have a great collaborator in Sohrab Shah, shown here, who has a team of informaticians who built the tools which enable us to take genomic information, whether it's RNA-seq information, tumor normal genomes, and to develop kind of lists of likely mutations, rearrangements, and to dig further and actually look at the mutational landscape and the clonal structure of tumors. The things which have been discovered are really exciting. So in the past, we knew about the home runs in cancer. Genes which were, if you're born with a truncating mutation, you might get cancer, or genes which look like viral genes, like KRAS. Well, these are sort of the home runs in the cancer game, but what really wins cancer, like in a baseball game, are the singles and doubles, and that's what we're seeing now. Change of function mutations, mutations impacting multi-protein complexes, modulators of host responses, and things like chromatin remodeling and developmental program gene mutations, which wouldn't have been apparent by any of the research which was done previously. We're also seeing features such as RNA editing and major changes in splicing in cancers, which need exploration, and how that will actually lead to improvements in care is yet to be seen. I study ovarian carcinoma, and our view has been that if we're going to make improvements in these sort of awful kind of survival figures, we're going to need to have subtype-specific prevention and screening strategies and be able to have subtype-specific treatments. But very little was known about these types of cancer, and therefore it was a great invitation to start doing some genomic analysis. We started by looking at rare ovarian cancers. The reason why is that whenever a new technology or way of thinking about disease has come along, a disproportionate amount has been learned from studying rare diseases. So you can think of Sir Percival Potts and these poor little chimney sweeps, the retinoblastoma gene, and Brenda Galley, who's been the international leader in this area, is in the room, and Burkitt's lymphoma and the role of viruses. So this has always been true. It's still true. So we took 4 granulosa cell tumors of the ovary. At this time, when we did this, there were editorials in journals saying you needed to look at 500 cancers to get knowledge rather than data, and we thought that that was rubbish, and so we went ahead. And these tumors are about 5% of ovarian carcinomas. At an early stage, they can be managed by surgery, but if they start recurring, there's no treatment. They also, in some cases, are problematic diagnostically, and so a new diagnostic is needed. We found the same mutation in the transcriptome in all 4. It turned out to be in a transcription factor, FOXL2, which is critical for the maintenance of the female state of gonadal stromal cells. So it was a very nice fit. We since showed that this is basically present in almost all adult-type granulosa cell tumors, not in other tumor types. And we developed an algorithm for how this could be used clinically. But before we published this, there was a lab in Brazil offering it as a diagnostic test, which we thought was pretty cool. We're also working on this, and we have all the constructs, and we're trying to figure out how we can better target tumors which have this mutation and hope to be able to translate that into some clinical trials. But the translation of a diagnostic into practice is quite easy. Treatment's a different story. So this is a mucinous carcinoma of the ovary. It's about 4% of ovarian carcinomas. And we showed that about 20% of these, so it's 20% to 4% of ovarian carcinomas, have massive amplification of HER2 expression. We can show it's a real skyscraper like amplicon by array CGH. We even have anecdotal evidence to show that women with this particular problem, they're very rare, have responses to trastuzumab or Herceptin. And so here we have a disease which is untreatable because high-stage mucinous carcinoma of the ovary is actually the worst of all ovarian cancers and outcomes. We have a drug everyone knows how to use. We have a lab test every lab can do. And yet we went to every clinical trials group we could think of, and they all said, hey, this is great, but it's too rare, we can't help you. And so I would propose that unless you can figure out how to do a trial like this, all the talk about personalized medicine is just hot air at this point, at least in the cancer domain. So we're going to try more of a social networking, anecdotal type approach where we're just going to work with interested physicians and forego the clinical trials process to test this at this point. Clear cell carcinoma of the ovary we got quite excited in because it's the second most common type of ovarian cancer, really not understood in any way, kind of a weird cousin of renal clear cell cancer. And we thought at the time, because it was reported to be genomically stable, so we thought a small study would yield some useful information. Here's the outcome showing it in early stage clear cell carcinomas. It actually has a very good outcome, suggesting what we may need here is some prognostic marker to determine maybe some people don't need any treatment at all and the surgeons are curing them, which would be great. But late stage clear cell carcinoma has an atrocious outcome compared to the more common high-grade serous cancers, suggesting here we desperately need new treatments. And so we went into a genomic process looking for different sort of clinically defined kind of endpoints. The first thing we did was sequence the transcriptomes on some cases as well as a cell line, and we found mutations in a gene called ARID1A. It's a part of the SWE/SNF chromatin remodeling complex. And we then got more tumors in from friends and family around the world and found that the gene is sort of plastered with mutations in a classic kind of tumor suppressor-like pattern. As a pathologist, when you ask the question, okay, how can you prove a mutation is pathogenic? Well, you can do the normal tricks. You can do a statistical approach. Do you see it in one biologic state and not another? Does it look in terms of pathogenic in terms of what it would do to proposed protein function? You can then do functional studies, or you can take more of a pathogenetic approach where we were able to track the mutation into the adjacent precursor lesions, which suggests that whatever it is, it's an early event. It's happened in the lesion before it even knows it's a cancer yet. We're then able to ask, well, Could ARID1A mutations be used to predict which patients with endometriosis will end up getting cancer? And atypical endometriosis is a rather difficult diagnosis. There's no proper criteria for it, but we managed, it's the most aggressive type of endometriosis, and it's the proposed bad actor. We only found 10 cases which we could access. One of the 10 had loss of ARID1A. We can show it. These are the epithelial cells, no staining, the lymphocytes still stain. And this was the only patient who got cancer, and she recurred with a cancer 2 years at the site of operation, suggesting that maybe there's some hope for this. It could be useful, but we need to work with others to move that ahead. So what we've done is we've got a project which starts with, we're going to sequence 50 clear cell carcinoma tumor normal genomes, RNA-seq, try to carry it through over a 7-year period into clinical trials. And we're kind of underway with this project. We got data from the first 10 cases, and another 10 are now almost finished the informatic process, and so we'll get those by midsummer. And the first thing we learned was rather shocking, is that we thought these were genomically quiescent, but they're actually pretty crazy. So here's a circus plot showing each of these cases, and for instance, this one here, there's an absurd number of amplicons and deletions in it. It looks just as bad as a high-grade serous cancer. Some of them are a little bit more quiescent. We also could look at all the mutations in these cases and start seeing, are there differences in the cases which have a lot of mutations and those with a few mutations. And when you get the next-gen sequencing data, if you validate the mutations at depth using other next-gen sequencing techniques, you can start determining the clonal architecture of the tumor, so how heterogeneous it is. For instance, this one here, we have a small number of mutations present in every cell and a bunch of mutations present in very few cells. And we were curious to see that the ones which had the greatest degree of clonal diversity seemed to be the ARID1A positive mutation cases, the ones with ARID1A mutations. And we're looking forward to getting data on more cases to determine if this is a feature. We're now taking all of our cases where we have, for instance, a clear cell carcinoma here, we have atypical endometriosis, and we have an endometrioid carcinoma coming from the same lesion, to sort of take the mutations from the tumors and getting a better sense of what actually goes on in endometriosis before it turns malignant. We can find things like PI3 kinase mutations in the endometriosis, but what's probably not that surprising, because it is appearing again and again and again in the literature in different contexts, is we find a lot of mutations which would lead to an aberrant inflammatory response being very, very early events in the genesis of these cancers. And so those who believe in eating flax oil and other things, it's probably, there's something to that here, at least from what we're finding. And we're looking forward to looking at more cases and doing more direct deep sequencing of endometriosis to determine how common these really are. So we've learned that some clear cell carcinomas have remarkable genomic disruption. ARID1A seems to be the boss gene. These are, endometriosis is much more complex than we thought. I think considering it a benign condition may not be really true. And the aberrant response to inflammation seems to be strongly implicated in the transformation of endometriosis into cancer. High-grade serous carcinoma is a completely different ball of wax. This was the subject of TCGA, probably come from fallopian tube. They all have P53 mutations, or almost all. About 50% have BRCA mutations. And this is what the chromosomes look like. They look crazy, the aberrant genomes. Post-TCGA, we got together as a group and said, what are the questions we want to address? Well, we want to look at what is the role of intratumoral heterogeneity and clonal divergence over time in these tumors, if they're so chromosomally unstable, because that's how everyone refers to them. We want to determine the relationship between functional and genomic heterogeneity and their relative roles in the emergence of drug resistance, and we're going to try to forge a collaboration with Ben Neal and his group to work on this one. And also what chromosomal instability actually buys a cell, which is really a question which needs answering. So we have this project, the structure and mutational heterogeneity of high-grade serous ovarian carcinoma over the space-time continuum, where we want to get multiple samplings of patients. We have many samplings at time of diagnosis and follow them through time. And just in case anyone in the audience doesn't think that people who work in labs are geeks, here is a picture from our first lab meeting. Now this is a very hot area, and AACR was just dominated by presentations on intratumoral heterogeneity. And it's interesting because in the pathology space, it means, well, how many times do you actually have to sample a tumor? And it looks in some incidences, our relationships with patients is going to expand into multiple samplings over time. There may be other instances where one bite can tell you everything you need to know for the path of that patient. Now this, of course, from studying tumors, multiple samplings of tumors, you can start teasing out the life history of that cancer and determining, well, what were the initial mutations? What was the precursor lesion? And it's a very exciting area. And technically, the next-gen sequencing is now emerging that there are ways of doing single-cell next-gen sequencing which can really kind of tease apart what the relationship between the cells in a tumor are. This is from the conclusion of a paper which says, hence, each patient's cancer may require individual-specific therapy, and even this may be thwarted by the emergence of a genetic variant, subline, resistant to treatment. And this is being sort of presented as the new thing, but that comes from Peter Knoll's paper in 1976. And this is another one of these sort of tumor evolution figures, but from 1976, and Isaiah Fiedler was working on it earlier. So I think this is even better than a new idea. This is like a road-tested idea that we're finally technically able to sort of deal with. And it's a very exciting thing to get into, and it's, as for pathology trainees in the room, you have access to the samples to do the appropriate banking to look at this. This was really shocking to us. So everyone says this is a chromosomally unstable tumor, it's crazy, it's drifting all over the place. But when you look at multiple samplings from the same tumor, the normals in the middle, they were identical. They're crazy, but they're not changing with time. Yet when you look at the mutations from exome sequencing, they're acquiring mutations though. So it's if it's sort of jumping track. The architectural structure of the genome is frozen in some new weird space, but it can still evolve in other ways. Here's a case where we have multiple samplings from different places, including actually a fallopian tube lesion, where we can actually start not just establishing the clonal relationships with putative precursors, but actually look at the direction of evolution to show it actually was the precursor. And again, here there are some changes with time. I think there's a little bit here you can see in the X chromosome, but these are remarkably similar. But you're still acquiring mutations at the base pair level. So here's, for instance, a mutation which is present in all the cells in one sampling, but is completely absent in some of the other samplings. So we're in the midst of doing laser microdissection on the precursor lesions. We fix the fallopian tubes in a special fixative so we have good quality nucleic acids to establish the relationship with the precursor lesions. And what we were really surprised is we went into this thinking that this was a very chromosomally unstable tumor. When we map out using the edit distance, all of the samplings from these different cancers, so each of the cases with multiple samplings all kind of cluster together, and they're in these very, very tight clusters. And the distance between all the normals from the patients is much greater than the distances between the tumors from any individual patient, suggesting these aren't really any more unstable than we are, I guess. So the early lessons have been a bit surprising, but high-grade serous genomes are very aberrant, but only slightly unstable. And this is a bit like me saying, I may be crazy, but I've always been this way, so I'm not unstable. So I can relate to this kind of issue. The cancers with apparent loss of chromosomal instability, so they're no longer evolving in this way, they're accumulating point mutations, and maybe when we look at the epigenome, we're going to see that they're moving rapidly upon the evolutionary stress of treatment. Some mutations are shared with precursors, and some are unique to regions of the cancer. And so it's an evolving story in our group, and there's groups in the UK and Australia working on this as well. So what are we really trying to do here by sequencing cancer genomes? Well, we want to identify features in the genomes of tumors that aren't apparent from morphological clinical assessment. We hope these features could be used as biomarkers to mark clinically distinct subgroups of cancer or as therapeutic targets. And we hope genomic analysis will often split cancers into new classes, like in the mucinous ovarian cancer story, where this is quite problematic because it creates very small groups of patients for stratified treatment approaches. But sometimes we can forge new groupings, and this is where it gets quite interesting. So for instance, if we see ARID1A loss and mutation in half of clear cell carcinomas of the ovary, a third of endometrioid ovarian cancers, you will see it in a much greater number of uterine carcinomas. And you also see it in 20% of gastric cancer, which is a very common cancer internationally. So if we figure out a way of treating tumors with ARID1A loss, it could be a very strange group of cancers which could go into that trial. We saw this in another story where we sequenced a bunch of rare cancers here, expecting we'd find a different type of mutation in each. And what we found was they all had occasional mutations in the RNase B domain of DICER, the microRNA processing gene. And it's an interesting discovery because it really challenges the way we think about how tumor suppressors actually act, because they're kind of hypomorphic alleles. We've been working out the function of these with my colleague Greg Moran, if we actually found a way of treating cancers with this mutation, again, you would have an absurd collection of different tumors which could be brought into that trial. And so it's tempting to think, though, that, well, don't ditch our old taxonomy of cancers and just start treating mutations. And this can work. We know that ALK fusions are targetable across multiple different diseases. KIT fusions are as well. Real HER2 amplification seems to be. But we know BRAF, it doesn't work, and PI3 kinase targeting is a real nightmare. And another potential challenge to personalized sort of medicine was raised in the last talk, and I think unless we get this right, the whole concept of genomic-based personalized cancer care could be really rocked through too many false starts. This is the BRAF inhibitor in melanoma, targeting mutant BRAF. It's a brilliant advance. The responses aren't durable, but compared to standard of therapy, it's a huge difference. The same drug targeting the same mutation in colon cancer doesn't work at all, because colonic epithelial cells can upregulate EGFR, which provides bypass. And so the context of where mutations occur is important. Finding a mutation isn't enough. So we're trying to pull this together in a model where we say, well, you can't just find a mutation in one cancer, treat it, find a mutation in another cancer and think the therapy is going to work. You have to consider other things. Like, is the mutation actually a driver in that cancer? Is it clonally dominant? What is the 2D and 3D mutational context? You also need tissue-specific in vitro validation, but for most drugs, that's already available through the Cancer Cell Line Encyclopedia. So finding mutation isn't enough. You actually can look in your data, if you have expression profiling, to actually say, is that mutation sort of warping the cellular kind of landscape in ways which you expect based on knowledge of that pathway? And so you can actually determine whether the mutation is a driver from the same data set which you find the mutation. You can also address the clonality issue. If a tumor is only present in 10%, a mutation is only present in 10% of tumor cells, you're going to have to argue that's a very special 10% of cells for that to be high on your list of targets. So you can now start teasing out what clonal populations exist within cancers. And if your mutation is kind of over here and it's present in almost every cancer cell, it's probably a much better target than it is if it's over here and present in only a very small number of cancer cells. Now, the sequencing of full genomes and exomes, there's a lot of challenges, and we may not be ready for that in the cancer clinic, but there are things you can do. We can go beyond looking at 1 or 2 mutations into some of these next-gen sequencing-based assays which look at several hundred mutations. And it's sort of the quality control issues for these massively multiplex issues have to be worked out. We have to work together as a community for how we're going to do that, but they offer great advantages over the way we do things right now. So some of the challenges, if we're going to go beyond those sort of massively multiplexed kind of assays, which are really just an incremental change into sort of full genomes or exomes, the issues which have been raised in a lot of the other presentations, the identification of reporting of features, how to deal with the longitudinal impact, and I learned so much from the first presentation today, which I really enjoyed. The challenge that no identifiable group of people knows really how to deal with the residual information. And there are broad-ranging concerns in healthcare, social, ethical domains, but actually these aren't problems. They're actually interesting challenges for people to study and embrace. However, given all this, it's hard to imagine that this isn't going to be our future. And in BC, we started to recognize whatever disease we're studying, the fruits of personalized medicine and doing genomics may be seen in a disease-specific context, but the challenges are shared no matter what disease you're studying. So we formed something called the Personalized Medicine Initiative, where everyone's been working together, and we're drafting a business plan right now, and we're engaged in some other activities. So we're creating an asset map for personalized medicine in BC. Probably BC isn't the only place where the government doesn't actually know all the things it's spending money on. We're establishing public policy for personalized medicine through work by Michael Burgess, who's using a deliberative democracy process to do this, identifying some frontline projects, looking into the tools and what capacity we need to link genetic and genomic information to health records. And again, I've learned a lot about this today. And we also want to establish model communities, small subsets of our population where we can determine the aggregate costs and benefits of personalized medicine solutions. So if this was the first, like 1850s, the disruptive technology in pathology, I believe this is the first disruptive technology since that time. And we go back to this kind of paraphrase. Now I'd like to introduce you to another well-spoken Canadian, Andre Picard, National Health reporter, who wrote a very provocative editorial in the Globe and Mail in this year, where he summarized and paraphrased reports of Justice Cregan and Cameron, who were discussing failures of diagnostics of all types in Canada. And since he could paraphrase those people, I'm going to take the liberty of paraphrasing Picard, who basically said pathology or diagnostic services in Canada are too screwed up even to consider supporting personalized medicine. This is a problem. But I don't actually see this as a problem. I think the things people have been trying to do in the past, making impossible diagnosis on glass slides, you can't. If you ask people to do something impossible, they're going to fail. I think genomics is digital, and it should be easier to do. And so with Susan Camel-Reed and others, we've decided to dig into this, and we've set up a national project where we want to determine whether the genomics tests we need to be done for tomorrow can be delivered in hospitals, in the Canadian hospital system, or if they can't, we're going to have to invent some sort of new laboratory system to deliver them. And so the goal of this project is to create and test a cooperative interprovincial approach to the evaluation and implementation of novel diagnostics. We're going to use a replicate testing approach using moderate complexity assays such as PAM50 breast cancer expression profiles, an ovarian cancer profile, and 2 massively multi-sex mutation screens in 2 small labs, 2 large labs, and 2 middle-sized labs. All assays to be done in hospital labs and compared to extant technologies and gold standards. And we have quite an elaborate kind of data analysis strategy which enable us to tease out whether the accuracy and precision of these genomic assays will exceed those of the extant technologies, and I believe they will. And I think that's why genomics will become dominant in pathology, because it's just easier to do than what people have been trying to do in the past. And so I'm not actually very pessimistic about things. I think it's a really exciting time in spite of Andre Picard. I think because I see what all these diagnostics are required from the point of target identification through to the monitoring of therapy. As a diagnostician, I realize I have the opportunity to define diseases not by just what they look like, but what they are. And I see a future for pathologists as omics interpreters to ensure that all studies performed on samples are summarized in single reports, and we'll still be able to serve our patients and their clinicians better. But I'm really uncertain whether the pathology community will be able to rise to this challenge, and this is a big worry, because we're not very forward-looking at the moment, and we should be looking forward how we can help our patients more by adopting this. And it may be we're not looking forward because we're overly worried about these characters, medical imaging and genomics, and there's turf stuff and control issues going on, because I think they're frightened that this may happen. But you never really know what can happen. And I don't understand this picture at all, really, but but the future's not always predictable, of course. And I see there's some great benefit in us thinking about, well, how can we best serve the patients tomorrow when you have all of these technologies converging? I think that pathologists and diagnostic imagers, informaticians, genomicists have to work together on cancer diagnostics, and I'm sure it's true in other areas as well. And I think if we do that, we'll be able to help our patients more. And because if you take the 2 kind of lions, genomics and medical imaging, and you cross them with a zebra, you actually get something quite magnificent. And so to any trainees, diagnosticians in the room, I got to say, you've got a great opportunity. You've got to be tigers here. You've got to start working with the other diagnostic specialties, and very importantly with genomics and informaticians. And I think if you do that, you might be able to create something which today's generation of diagnosticians could never really imagine, and you'll be able to help your patients in unbelievable ways as well. At this point, I'd like to thank you for your attention.
We have time for some questions. So wonderful talk. Thank you very much. David, the tumors in the clear cell carcinomas that were genomically unstable, did that at all correlate with grade? the extent of instability?
Well, the problem with clear cell carcinomas, there's a kind of they're de facto always called high grade. So they're always just called grade 3 as part of like the clear cell carcinoma, call it grade 3, which actually doesn't make any sense. We are going to go back and look if they have any particular features. We also are going to determine, because we have access through a program called the TFRI Cure, where we have a cohort of 2,000 ovarian cancers. We're going to have outcomes to determine whether this genomic instability in clear cell carcinomas is indicative of bad acting tumors, because we have a feeling it may be from some other kind of rather loose data.
I guess I'll ask one more question then. You've really put up the challenge for pathologists. It's an exciting time. I was also intrigued by your comment about the endometrioma and the changes that you're seeing. Would the other way to look at those alterations be that we can get these changes and the significance of them are really not known yet?
Well, we haven't seen similar changes in endometriosis not associated with cancer. So that would suggest that this is, this is anyway associated with cancer. And endometriosis has not really been much studied as a cancer precursor. And only a very small number of endometriotic lesions will go on to develop cancer. They're usually endometriomas in the ovary. And I'm really intrigued that so many of the early events tie into sort of aberrant inflammatory responses. And what that's telling us about what's going on inside an endometrioma, which is some sort of toxic, kind of awful hypoxic place. And we're looking forward to working on that. And there's an excellent mouse model for endometriosis, which was developed by Steve Charnock-Jones in University of Cambridge. And we're starting to insert our mutations into his model to see what happens.
Survivorship of ovarian cancer was 32%, all invasive. In 2003, the survivorship was 32%. We've made no progress in curing ovarian cancer. I'd say since surgery was introduced, certainly for 40 years, despite the introduction and dissemination of widespread use of chemotherapy, Taxols, platinum, cisplatin. There has been an advance in extending the disease-free survival, but no advance in 40 years in curing patients. Essentially, the patients that are going to recur have all recurred and they will recur. And you can prolong that or prevent that for a short time, but not ultimately. And to me, that's a kind of a limitation that we have to consider as the standard is the cure rate, not the 5-year survival rate. Is there anything that you think that in the personalized medicine approach can overcome that?
Well, I think there are some ovarian cancer patients with low-stage clear cell endometrioid and mucinous carcinomas who are receiving overtreatment. They're likely being poisoned for nothing. There are some carcinomas like high-stage mucinous, high-stage clear cell carcinomas where the treatment we give has no impact at all. They should probably go straight into trial. There are some intermediate-stage clear cell carcinomas which seem to respond to radiation therapy, and we have data to suggest that that may be the best option for them. For the high-stage serous carcinomas, which are the dominant disease, it does suggest that we need if we want to have at some point to have cures, we have to have a complete rethink of the way we're managing this. And because gradual extensions of disease-free survival with no overall survival benefit isn't really progress you can be proud of.
Well, we do always talk about, as the goals of personalized medicine, to avoid giving treatment to people where it doesn't work, which is a fine goal, but it's not the one we seek, which is to cure patients that we're not curing now. I mean, how much of this is, I read an awful lot about people who don't need chemotherapy, and I read very little about new chemotherapy that may actually work.
I think with high-grade serous carcinoma, I actually believe this is, it didn't break down. So TCGA's job was to break this into a bunch of distinct diseases. And they applied every tool in the world, and it wouldn't break. in a meaningful way. So I think this is a disease there could be big solutions. And the combination of BRCA loss and P53 mutation, which is sort of a dominant feature and leading to chromosomal instability, is very strange that fallopian tube epithelial cells, they can somehow survive this insult and become a cancer. Some type of breast epithelial cell can as well, but in the rest of the body, not so much. So there may actually be intrinsic features of the cells of origin which could be targeted in ways which make the genomic unstable state intolerable. So I think there's either that or immunotherapy. There's going to be changes, but it's not going to be from tinkering with in the same sandbox we're in right now. Yeah. Thanks.